the...
I'm better off reteaching it.
Wait.
I'll do it after.
Oh, okay.
Remind me, okay?
Sure.
Okay, good. Okay, shh. Just a quick reminder, erythrocytes, which, by the way, what are erythrocytes?
What?
Red blood cells is correct.
Oh. I remember.
Apologies for the interruption. Now or when most convenient, could we please have Mr. Cooper in guidance?
Sure, yeah, absolutely.
Thank you.
So, do you need to go to guidance?
He said now or whenever.
Perfect.
This is the first time that I'm excited.
That was really soft, though, right? Was that really soft?
No.
She's like, I'm sorry to interrupt you.
Yeah, like, come on. Let's have some volume here.
Sorry to interrupt you.
Orlando's ears aren't getting any younger, right? Okay, so we have little markers on the outside of the cell surface, and like obviously this is just schematic. I'm not suggesting they actually look exactly like this, but these are called antigens, all right? Antigens, which are basically like markers, or you can even think of them as like flags. These are usually made of some combination of a chain of sugar molecules, so like carbohydrates, and sometimes with lipids. The specific marker structure, that's actually a good, not something I've looked up in decades. This, by the way, is why I get off track every lesson. A antigen structure.
Antigens for me, because all I can think about is them actually having flags on them.
But what they actually are is they look like this, where they are, I mean, even that's not a great look.
Color coded?
Yeah, well, we'll come back to it. But yeah, they're what we call glycolipids. So they're made from a combination of sort of carbohydrate chains and fatty acid chains. And they serve as flags on the surface of a cell. These are binding sites. Right? They're binding sites. What does something mean if it's a binding site? What does it mean to bind to something? To get together, to stick together, yeah, absolutely. You can stick to something. In other words, an antigen serves as an identifier, and it's a place where something can stick to it. Does that make sense? Okay. Sorry?
That's binding, right?
Yeah.
Okay.
I will do that again. Now, as you recall, on chromosome number 9, we have a gene. I'm going to draw chromosome number 9. Just as I guess we'll draw it like not in its duplicated stage, because why would we? There's chromosome number 9, and right here is the ABO gene. And if you recall, there are 3 genotypes of the ABO gene, or there's 3 alleles, I should say. Remind me, what's an allele? It's like a version of a gene, right? So this is the gene that codes for ABO antigens, right? So in fact, maybe we'll take a second and say this. The gene codes for the production of antigens. There are 3 alleles. The first allele is A, and that codes for A antigens. The second is B, and it codes for B antigens. And the third is O, and what does it code for?
O antigens.
Other O antigens?
No, it doesn't code for anything.
Yeah, it codes for nothing. And if you recall, these are both codominant, which means that they can both be dominant, which means if you have A and B, you'll have A antigens and B antigens. And the O we think of as being recessive because you wouldn't have no antigens unless you have 2 copies of the O allele. What are the possible phenotypes? You could be AA, AB, BB, or
BA.
we've got those.
Oh wait, BO.
Yeah,
AO.
yeah,
OO.
good. Why did I put these together? Yeah, because A is dominant, so you'll be type A whether you have AA or if you have type AO. Either way, you will be blood type A. And what about here?
Type AB.
Type AB, very good. And here?
Type B.
Type B. And here?
Type O.
Type O, good. And I'm going to use, what am I going to use? I'm going to use blue circles for type A. Okay, so look at me. I've got a whole bunch of type A antigens on me, which also means type AB is going to have a whole bunch of blue circle antigens on me. Okay?
Bentley Harris, please come to the office.
And I'm going to use green squares for type B. So type B has got a bunch of green squares on it. Oh, look at me. I've got all these green square antigens, which again are binding sites, which are cell markers. And it means if I'm type AB, I've got both of them. Does that feel comfortable?
Yep.
And again, if I'm type O, what do I have?
Nothing.
Nothing. Nada. Bubkiss. We don't need to write this much, but we'll just say, since A and B are both dominant, there are 4 possible phenotypes. Okay, and then we know that there's actually a third antigen that we can have. So why didn't we include it in this discussion? What's the third antigen? There's the A antigen, the B antigen, and RH. Yes. The RH antigen. I feel like there was a pirate joke in the making there. If I was a punnier teacher, I'd have, I don't know. I'd be like, what's a pirate's blood type? They're all RH positive. I don't know. I don't know. See, this is why I don't do this. Because the thing about puns is I find them annoying. But the truth is, you also have to be quite clever to make puns. And so then I wonder, like, is my dislike of puns just sour grapes? Do you know the expression sour grapes?
No.
Like jealousy?
Yeah, well, kind of. Sour grapes isn't just jealousy. Sour grapes is specifically when you like say something negative about something as if you didn't want it, when really it reflects that you are jealous that you don't have it. So it comes from a fable about like an animal that isn't able to get these grapes, and they say like, oh, they're probably just sour grapes anyway. But they don't really mean that they think the grapes are sour. What they mean is that they're jealous that they didn't get the grapes, right? So I'm like, I think puns are stupid. But maybe really what I mean is I think I'm stupid and I should be better at puns and I don't get them. You know what I mean? Mm-hmm. So there's some insecurity there. You can draw your own conclusions. Let's move on to the RH factor. Okay. The RH factor is another antigen controlled by an entirely different gene. Did we say why it's called the RH factor?
I think we did, possibly. This
is apropos of nothing. I'm going to write in blue. You have no reason to write this if you don't care. but it is called Rh factor after rhesus monkeys
Is that entirely?
in which it was discovered, in which they were discovered. But we have the same factor. I feel like I've misspelled that, but that's okay. We'll look it up after. You don't need to know that. That's not important information, but I think it's kind of cool.
Is that an R next to the I? In like that's entirely correct.
This?
No, that. Okay.
Just who cares? Just write it. I trust you. Okay.
Okay.
So now there's two alleles, and the two alleles are Rh positive and Rh negative. Does that make sense? And the Rh positive allele is dominant, and the Rh negative allele is recessive, which means in the end we have two phenotypes. You can be Rh positive or you can be Rh negative, but there's actually two ways to be Rh positive, which are what? You could have two copies of the positive allele, or you could have one copy of each. And in either case, you'll have the Rh factor, which I'm going to show as a red triangle. I really feel like this lesson lets me engage with my inner kindergarten teacher who just wants to draw colors and shapes.
Can you scroll a little bit up?
Yes. And what would the genotype have to be for someone to be Rh negative?
RH negative.
Rh negative,
RH
Rh negative. They have to be homozygous recessive. Good. If they are heterozygous or homozygous dominant, they'll be Rh positive. Your blood type is a combination of your ABO phenotype, A, AB, B, or O, and your Rh phenotype, positive or negative. So how many possible combinations? Altogether, there are how many different blood types? A combination of those and positive or negative. So you could be A positive or AB positive or AB negative, B positive or B negative, or O positive or O negative. So how many? So there are 8 possible blood types. Since there are 2 traits, involved, tracking heredity requires a dihybrid cross. What the hell does that mean? Tracking heredity requires a dihybrid cross. What kind of question was that? You guys did very well on this on your previous unit test. Good old-fashioned 4x4, right? You could be like A, B, R positive, and this would be AR positive. You don't have to actually draw this, but here we are. And this person could be AO, R positive, R negative, and then we'd be like AR positive, AR negative, OR positive, OR negative, and so on and so forth. That's just an example. Do you need to draw that?
No.
Absolutely not. Yeah. Sorry? Tracking. In other words, if you're going to track the heredity of blood types, you've got to track both the ABO gene and the Rh gene. Why is blood type important? Why is blood type important? Yeah.
If you're donating blood, you need to have the same type as somebody.
Yeah. Why? Why does that matter?
Can't you get severely ill?
Because the blood can attack the other blood cells.
Very good. Remember, we learned about blood types, and what's one of our blood cell types?
The white.
The white blood cells, what do?
Attack viruses.
They attack anything that is foreign, right? Foreign cells come in, your white blood cells' job is to attack. Well, wait a minute.
So they
What if you have blood cells that don't look like they're yours? Then what's going to happen?
cannibalize the blood cells.
Now, how do your white blood cells know what blood cells are there? Because of little flags. That's what they look for. So, the immune response. Your leukocytes. Yeah. Am I going too fast? I'm sorry. I'll slow down. Can we all read that?
Oh wow.
You don't need to draw this. I would not bother drawing this.
Okay.
I drew it so we knew what I was talking about.
Yeah.
And if you really feel like it, you can draw it, but you don't really need to draw
I
it.
just
Right?
noticed you had the pencil thing back.
Yeah, I've downgraded. Right now, I'm working on the older version of OneNote, which has some annoying features to it, but I have my pencil back. I need my pencil.
I like with the pencil.
I like
That's
right.
nice.
Isn't that nice? You can shade
Yeah.
and you write with the tip of it, and it's that, and you write with the side of it, and it gets nice and wide. Yeah, I like the pencil. Anyway, that's about me, my pencil. Oh, these are filthy. Put my stupid sweaty hands all over them. Okay, so we are going to move on now. Your leukocytes, your white blood cells, are designed to identify and destroy foreign cells. I shouldn't say designed, I should say specialized. See how easily we slip into anthropomorphic language. Are specialized to identify and destroy foreign cells. This includes incompatible blood cells. Please. This is an important thing to know. Before we really fully understood this stuff, they knew that you could give one person's blood to somebody else. They also knew that sometimes that was successful, and sometimes it killed the person real quick, right? And that's not good. And then as we figured out blood typing, we figured out ways to avoid killing people real quick with foreign blood, which is good. So the trick is, of course, what's the only way that your body can identify foreign cells? It needs to be on the lookout for foreign. What part of the cell? The little spiky flaggy bits, which are called antigens. Enter antibodies. Now, do I like that antigens and antibodies both start with the word anti? Interestingly, antigen is antigens are produced by the body in response to foreign antigens. In fact, the word antigens literally means antibody generators. Does that make sense? Genesis means, what does Genesis mean? First book of the Bible. It deals with creation. Very good. So to create, and anti in this case just means referring to antibodies. So antigens literally are the things that induce your body to generate antibodies. So these make your body produce or generate antibodies. So the antibody itself are an integral part of your immune system. These are these identify and bind to non-native antigens. And there's 3 types. You can have anti-A antigens, which bind to A, right? Anti-A, if it sees A antigens, and your body isn't supposed to have A antigens, it will bind to them, and then it will signal the immune response. Does that make sense? We have anti-B. These bind to B antigens. And finally, we have no, O's nothing. O isn't an antigen. O is just a lack of an antigen. So the last one we have anti-A, anti-B, and what's the third antigen? No, that's just having A and B. What's the third antigen?
Nothing.
No.
Positive and negative?
Yeah, which we call the RH factor. Because the positive just means it has it, and the negative just means it doesn't. So we have the anti-RH factor, which binds to the RH factor. Once an antibody binds to a blood cell, it starts sending signals like crazy. Hey, body, send the white blood cells here. Let's get destroying this stuff. Does that make sense? So if an anti-A antigen binds to an A antigen, it's going to signal your body, come destroy these cells, and then your white blood cells are going to come and destroy all of the blood cells that have just been put in you. But because that's a big, massive amount of cells, it will literally just turn that part of the blood into a big coagulated mess that will cause a blood clot, and the person will die. Does that make sense? So here's the type. Here's the thing to know. I'm only going to do one of these. You're going to fill the rest of this table in yourself. Maybe I'll do 2 of them. Let's say that you are type AB positive. In that case, what antigens does your blood cell make? If your blood type is AB positive, what antigens are on each of your blood cells? The A and the B, and what's the positive mean? RH. Now that means your body, are you going to make anti-A antigens that would kill and destroy any blood cells that have the A antigen? No, because if you did, you would kill your own blood. Does that make sense? Is your body going to make anti-B antigens? No, because then it would destroy its own blood. Is your body going to make anti-RH antigens? No. So if you have AB positive blood, your blood will contain no antibodies because your blood does contain all of the antigens, which means you can safely receive any blood. Does that make sense? If you have type AB positive blood, because your body is used to seeing the A antigen and the B antigen and the RH factor, there's no blood cell you can get that would be foreign to it, and so you can safely receive any blood. Does that make sense? Your body doesn't produce any antibodies because any antibodies your body produced would destroy your own blood. Does that feel okay? Okay. What if you're AB negative? In that case, what antigens would be present on your blood? If you're type AB negative, the A antigen, the B antigen, would you have RH? No, because you're RH negative, which means what antibodies would your blood produce? What would it be on the lookout for? It would be on the lookout for anti-RH, which means who can't you get blood from? Yeah, you can't receive positive blood. So you couldn't receive A positive, you couldn't receive B positive, you couldn't receive AB positive, and you couldn't receive O positive. Any of the above would be deadly to you. Does that make sense? Mm-hmm. Because if that positive is present, you have anti-RH antibodies, your blood is going to see that blood and you're going to destroy it. Do you feel like you could fill out the rest of this table on your own? Or do you want to do a couple more? You want to do a couple more? Give me the next blood type we should look at. B, okay, that seems like a weird alphabetical order to me. What maybe would be a more logical one? A might make more sense.
Yeah.
And do we want to do A, since we did positive first here, should we do A positive first?
Sure.
No.
Let's
Sure.
just go with that. What antigens would be on your A positive blood?
A.
A and
RH.
RH. Which means what would your body be on the lookout for?
B.
So you're going to make what we call anti-B antibodies, which means you can't receive what blood?
A, B, E.
You couldn't receive AB, right? Positive or negative. What else?
A, BO.
Which would just be B.
yeah.
And you wouldn't be able to receive B positive, but you also wouldn't be able to receive
B negative.
B negative. Does that make sense? In other words, doesn't matter if it's positive or negative, doesn't matter if it has A or not, but what's going to be deadly to somebody with A positive blood? Anyone that has a B in it, right? Okay, what if you're A negative? What antigens would be on your blood? Just A, which means what antibodies do you produce?
B.
Anti-B and anti-RH. Man, who can this person get blood from? Can't get what? Well, you couldn't even get A positive, right?
No.
You couldn't get AB positive. You couldn't get O positive. And you couldn't get B positive, right? In other words, all of the positives would kill you, just like this person, right? Because the RH factor is going to get you. But what else can't you get?
B positive.
You couldn't get B, we already have B positive, but you also couldn't get B negative, and you also couldn't get AB negative, right?
Yeah.
Would O positive, would O negative be fine? Yeah, because O negative doesn't have RH on it or B on it. Would A positive be, would A negative be fine?
Yes.
Yeah, but the only people that somebody with A negative blood can get blood from is a fellow A negative or an O negative. Does that make sense? They can't get any blood that has a B in it or any blood that has a positive in it. So if you have type A negative blood, your options are more limited than if you have A positive blood. Who can receive blood from anybody? AB positive. Real advantage, right? What should be the next blood type we look at? B, and we could either look at B positive or B negative. If you have B positive blood, what antigens are present on your blood cells? B and RH, which means you're making what antibodies? Anti-A. Just anti-A, right? Which means you can't receive what?
A positive, A negative.
We can't receive A positive. We can't receive A negative, AB positive, or AB negative. Again, what's the problem here? Doesn't matter if it's positive or negative. The problem would be if you were getting A. And the reason you can't get A is because your body produces anti-A, which will be on the lookout for A, and if it sees it, will destroy it. What if you're B negative? What antigens do you have? Just B, which means what antibodies are you producing? I heard anti-B, and I heard anti-RH, and that sounds good to me, which means who can't they get from?
Wouldn't it be anti-A?
Did I say B? Sorry about that. Somebody said B and I just jumped right along. You tell me what to do, I do it. You could say like, hey Orlando, do you know that your wizard name is your credit card number and its expiry date? And I'd be like, oh, that's a weird wizard name. Anyway, how's this sound, guys? Just dumb like that. Anyway, who can't they receive from? They couldn't get from A positive. Who else? A negative. Who else? AB negative or AB positive. Who else couldn't they receive from? That takes care of everything to do with A, but they also couldn't get O positive, and they also couldn't get, what's the other positive one they could get? The other possible B one would be, or positive one would be B positive. Even though they're type B, they can't type B positive because what will get them? The Rh factor. Can you see it? This is like important business because, yeah. Okay, who's the last ones that we have to deal with?
O.
And there's two types of O actually. There's
O positive.
O positive and O.
Negative.
Now O negative we'll do first actually. O negative has no antigens whatsoever on their blood cells, right? Which means what is their blood on the lookout for? Yeah, it will produce anti-A antibodies, but it will also produce anti-B and it will produce anti-RH, which means who can O negative receive from?
Only O negative.
Yeah, it can't get A positive, A negative, B positive, B negative, AB positive, AB negative, or O positive. The only person it can receive from is O negative itself. If you have type O negative blood, you can only receive blood from a fellow O negative. On the other hand, if you donate blood as O negative, have we come across a single person who can't take your blood yet? No. Because what doesn't your blood have? Antigens. And why can blood be dangerous? Because the cell is on the lookout for the antigens. So we often call O negative the universal donor. If somebody comes in from an accident and they don't have time to plan or be sure what their blood type is, what blood type do they give somebody? O negative. Does that make sense? Yeah. Sorry? Yeah, be quick, please. So on the other hand, O positive, what antigens do they have? They just have the Rh factor, which means that they will produce anti-A and anti-B. And that means that you won't be able to give them A positive or A negative, B positive or B negative, right? Or AB positive or AB negative. But the good news is you will be able to give them O positive and O negative, right? Because they don't care about the Rh factor. So if you're O positive, you can at least get blood from two types of people. If you're O negative, you can only get blood from somebody else who is O negative. So you think of like people who have O negative blood are incredibly giving, I guess. They're the universal givers. And on the other end of the spectrum, AB positive, what's true about them? So what do we call them?
Takers.
Not taker, though I guess we could. We call them the universal recipient.
Wow, recipient.
If you have type AB blood, you can receive blood from anyone. If you have type O negative blood, you can donate blood to anyone. And anywhere else in the middle, you have to be mindful. We're going to do a little lab later on. It's not like a big marked lab or anything, but it's just a fun little activity. We might do it tomorrow, depending on my mood.
I don't know my blood type.
Yeah, who does know their blood type? Mine's easy to remember. I'm type B positive. I think of it as instructions. I think my blood is just telling me how to think.
B
Be positive. Yeah. O positive? O positive. There you go. Fair enough.
I should ask my blood type.
Yeah, your parents may not even know. I don't even know they test anymore. There's some homework questions here. A really interesting question is, you could do a diet, but look, this is actually a really interesting thing. Is it possible for a mother to have a different blood type than one of her children? Is that potentially dangerous?
Yes.
Now, do mom's blood and baby's blood normally mix?
No. I
Do you know this?
I don't think they do.
No. In fact, like, mom has a lot of blood near baby, but baby is held inside, no, not inside the placenta. Yes, inside the uterus, but inside the amniotic sac, right? Surrounded by amniotic fluid. So baby's body is physically separated from mom's body, and then the placenta is not part of mom's body. The placenta is actually part of baby's body and interfaces with the endometrial lining. So there's big massive blood vessels that forms on the endometrium, right? That forms on the wall of the uterus. And those interface and they pass things back and forth But in theory, no blood from one should enter the other, right? They should be separate. But if they do, this is actually highly important. If even a little bit of blood is transferred, that pregnancy actually, there won't be an immune response because it takes the blood for a small amount of a foreign invader. It can take the body a while to matter. But if mom's body becomes sensitized to her first baby's blood type, by the time she gets pregnant a second time, there's a risk that her body will produce antibodies that will actually destroy the fetus the second time around.
Oh.
This is a thing that we can prevent with using something called Rogan or Winrow. And they don't even, if a mother, this is specifically dangerous with the Rh factor. For whatever reason, those antibodies tend to be more aggressive and tend to more actively cross the uterine lining. And so if an Rh negative mother is having a second child, they automatically just by default now treat her with Winrow. They don't check to see dad's blood type, especially because you never know. You know, people aren't always sure who the father is or being honest about who the father is or whatever. And so they just by default now will treat an Rh negative mother with this thing called Winrow to ensure that her body isn't producing antibodies that will destroy the fetus. Anyway, so there's some good questions here from homework. You can look these up. Shouldn't take you all that long, and I will commend it to your reading. And then